about the 4 levels of protein structure and my protein, which is histones. So I want to start off by talking about the 4 levels of protein structure. This would be the primary structure. This is really just the chain of amino acids in the order of them in a polypeptide chain. And then this is the secondary structure. This is the folded structure that they form inside the polypeptide chains. And then we have the third structure, which is the tetrary structure. And this is the 3D structure of a polypeptide, and it's formed from interactions between R groups and amino acids that make up proteins. And then this is quaternary structure. This is the 4th structure. This is formed by multiple polypeptides, and is the structure of a protein macromolecule. Okay, then I'm going to go on. So this is how a histone is essentially made up. There's different, histones are made up of arginine and lysine. They're pretty much the basic proteins that wrap around DNA, and they are able to help the DNA condense in order to fit into the chromatin. They are positively charged, which allows them to interact with the protein because the protein is negatively charged. It's negatively charged phosphate groups. So in order for a histone to be formed, I would say this would be the primary structure, but I don't think it necessarily fits into the regular realms of what the primary, secondary, tertiary, and quaternary structures of proteins are. So, but this would be the primary, but it's not an amino acid or anything like that. So I wouldn't say that it necessarily falls into that pattern. So we have the H2 markers and H2B markers, which go into the H1, and then we have H3 and H4, and those combine, 2 of those combine to make, well, 2 of each of these will actually combine to make the primary structure of the histone, the H1, which is used in DNA. So here's a little bit better image of what is happening. So the DNA is wrapping around those histones because it's negative, and these are able to combine because of the charges on it to create nucleosomes